Welcome back to day 35 of our deep dive into receptor tyrosine kinases, or RTKs. It's been a long journey, but it all culminates today as we look at therapies that directly target RTKs.
It's exciting to get to this point. We've gone over so much in this deep dive, and now we're at the forefront of medical innovation.
Right, so we're all on the same page. RTKs are often implicated in diseases like cancer because they drive that uncontrolled growth.
Yes, exactly. Uncontrolled cell growth.
So how do we apply the brakes? How do we use what we know to actually target these things?
Well, it's all about disrupting or modulating that uncontrolled signaling. That's where targeted therapies come in. Specifically, there are two major drug categories we should talk about. Tyrosine kinase inhibitors, we call those TKIs, and monoclonal antibodies, or mAbs.
Okay, so you've mentioned TKIs and mAbs before.
Yeah.
Could you remind us how they work?
Sure. So think of TKIs as highly specialized wrenches that fit perfectly into the active site of a specific RTK. They prevent that RTK from functioning.
Ah, okay, so by blocking the activity of the RTK, they essentially halt the transmission of the growth signal.
Precisely.
Okay, and what about mAbs? How do they differ from TKIs?
So while TKIs work intracellularly, mAbs are more like guided missiles that target the RTK from the outside.
Okay,
They bind to the extracellular portion of the receptor, which either prevents the binding of signaling molecules or it flags the cell for destruction by the immune system.
so it's a two-pronged attack. TKIs disrupt the internal machinery, and then mAbs interfere with the outside signals.
Exactly. And what's even more impressive is that these therapies can be so precise,
Good.
they can actually target very specific mutations in these RTKs. So we're talking personalized medicine.
Wow, so it's not just hitting the brakes, but it's like hitting the brakes on the exact part of the car that's malfunctioning.
Exactly. A great way to put it. So for example, in lung cancer, a TKI might be designed to target a specific mutation in the EGFR receptor. And then in breast cancer, a mAb could be engineered to target HER2.
That's incredible. But I remember we also talked about the challenge of resistance. Even with targeted therapies, cancer cells can adapt and escape treatment.
Unfortunately, you're right. It's this constant arms race. Cancer cells are incredibly adaptable. They can develop resistance through many different mechanisms. For example, the targeted RTK could mutate, changing its shape so the drug no longer binds effectively.
So even with these precise therapies, there's a chance that the cancer cells could outsmart them.
It's possible. And another way resistance can emerge is through the activation of alternative signaling pathways. Think about a city with multiple roads all leading to the same destination. If you block one road, traffic can just take another route.
Ah, so the cancer cells find detours.
Exactly. They can activate bypass pathways circumventing the blocked RTK and continuing to receive those growth signals.
Okay, so that's the problem. What can we do about this?
Well, that's where the newer generation of therapies comes in. One promising approach is bispecific antibodies. So imagine an antibody with two arms, each one capable of binding to a different target.
So instead of blocking one road, we set up multiple roadblocks.
Precisely. This makes it much harder for those cancer cells to escape. For example, a bispecific antibody could target two different RTKs involved in the same pathway, or even an RTK and another protein involved in immune evasion.
Oh, very clever. I've also heard about antibody-drug conjugates.
Ah, yes. ADCs.
What are those?
ADCs are another fascinating approach. Imagine a guided missile, but this time, instead of just blocking a target, it actually carries a toxic payload directly to the cancer cell.
Wow, so it's like a targeted airstrike.
That's one way to think about it. The antibody part acts as a homing device, delivering the drug right where it's needed.
Okay, I think I see where we're going with this. We've got all these powerful single agents, but as you've explained, cancer cells can become resistant. This is where combination therapy comes in, right?
Absolutely. Using multiple drugs that target different parts of the signaling pathway or use different mechanisms can be much more effective than just a single drug. It's like attacking an enemy from all angles.
So it's not just about hitting the brakes, but also cutting the fuel lines, disabling the engine, and throwing a wrench in the gears, all at the same time.
That's a great analogy. And combination therapies are already proving to be incredibly effective for many types of cancer.
Okay, I'm really excited about this. What about the future? What's next in the world of RTK therapeutics?
Well, the future is wide open. We're discovering new RTKs all the time and unraveling their roles in various diseases. And this understanding is going to pave the way for even more targeted and effective therapies.
So we're expanding our map and designing even more precise therapies.
Exactly. And we're also seeing these cutting-edge technologies like gene editing that could allow us to actually manipulate RTK activity at the DNA level.
Wow. So we're moving from just hitting the brakes to redesigning the engine.
Right. The potential of RTK therapeutics is vast. We're only just beginning to tap into its power.
This has been fascinating.
Yeah.
Before we move on, what are some key takeaways for our listener?
Well, the main thing is to remember that RTKs are these pivotal players in cell signaling, and their dysregulation can contribute to a wide range of diseases. We've discussed the major drug categories targeting RTKs, TKIs, and mAbs. They're both powerful, but susceptible to resistance.
And we've seen how scientists are tackling that challenge with new therapies like bispecific antibodies and ADCs.
Yes. We also talked about the importance of combination therapies to overcome resistance. And finally, we took a look at the future, where new discoveries and technologies are expanding what's possible with RTK-based therapies.
This has been incredibly enlightening. So what's next on our agenda?
Well, let's shift our focus to a specific class of TKIs. They're known as BCR-ABL inhibitors, and they've really revolutionized the treatment of CML.
Okay.
We can explore how they work, the success stories, and the challenges that still exist.
Sounds good. Let's dive in.
BCR-ABL inhibitors are just a remarkable example of how we can use molecular understanding of a disease to develop these truly transformative therapies.
Okay, I'm interested. Tell me more. What exactly is BCR-ABL and how does it cause CML?
So BCR-ABL, it's a fusion gene. It's created when two chromosomes, 9 and 22, break and swap pieces. This swap fuses the BCR gene from chromosome 22 to the ABL gene from chromosome 9.
So it's like taking two separate instruction manuals and combining them to create this hybrid set of instructions.
That's a great way to put it. And this hybrid BCR-ABL gene produces a fusion protein that acts as a constantly active tyrosine kinase. And remember, tyrosine kinases are enzymes that are critical in cell signaling.
Right, and especially in regulating cell growth and division.
Exactly. So in CML, this BCR-ABL fusion protein sends these continuous signals that tell bone marrow cells to produce too many white blood cells.
So it's like a factory that's been reprogrammed to produce way too much of one product.
Exactly. And before we had BCR-ABL inhibitors, CML was often a fatal disease. But the discovery of this fusion gene and its role really opened up a whole new avenue for therapy.
Okay, so how do these BCR-ABL inhibitors work? What's the mechanism?
Okay, so these inhibitors, imatinib, dasatinib, milatinib, they're all designed to bind to that BCR-ABL fusion protein.
So they're like specialized wrenches that fit into the active site of this specific fusion protein.
Precisely. And the results have been truly remarkable. I mean, since the introduction of imatinib, which was that first BCR-ABL inhibitor, the survival rates for CML patients have increased dramatically.
It's called a revolutionary breakthrough.
It really has. These therapies have turned CML from a deadly disease into a chronic condition.
That's incredible. But you mentioned challenges earlier. Are there still obstacles in treating CML?
Well, while the success of these inhibitors is undeniable, there are still challenges. One big one is resistance, just like we talked about before with other targeted therapies.
Right. Even with these drugs, cancer cells can still adapt and escape.
That's right. So how does that resistance develop in CML?
Yeah, how?
Well, one of the main ways is through mutations in the BCR-ABL gene itself. These mutations can change the structure of the fusion protein so that the inhibitor doesn't bind as well.
It's like the cancer cells change the locks.
Exactly, and there are many different mutations that can cause resistance to different BCR-ABL inhibitors.
So it's like they're always trying to stay one step ahead.
Right, always evolving. So how do we address that? Well, one approach is to switch to a different BCR-ABL inhibitor if resistance develops.
Okay.
So, for example, if a patient becomes resistant to imatinib, their doctor might prescribe dasatinib or nilotinib. Those target the fusion protein in slightly different ways.
So it's like having a backup key.
Exactly. Another strategy is to use combination therapies. Hitting the cancer cells from multiple angles can make it much harder for them to become resistant.
Okay, that makes sense.
Yeah.
Identify the problem, target it, and then find ways to overcome resistance.
You got it. It's a constant battle, but with each new discovery in therapy, we're getting better at treating this complex disease.
This has been a really insightful look at BCR-ABL inhibitors. Any key takeaways for our listeners?
The main point is that BCR-ABL inhibitors are a great example of how targeted therapies can completely change how we treat cancer. By targeting the molecular driver of the disease, these drugs have transformed CML from a death sentence to a manageable condition for many patients. But we still have challenges, especially drug resistance. So ongoing research is focused on overcoming these obstacles and developing even better therapies for CML.
This has been fascinating. It's amazing how understanding a single protein, the BCR-ABL fusion protein, has had such a huge impact on the lives of so many people. So what's next on our RTK journey?
Let's move on to another interesting area, the role of RTKs in the nervous system. We'll explore how these receptors contribute to things like neuronal development and synaptic plasticity, even things like learning and memory.
I'm intrigued. Let's dive in.
You know, it's amazing to think that RTKs, those receptors that we usually associate with cell growth, are also critical for our nervous system.
Okay, so how do receptors that are known for driving cell division contribute to something as complex as the nervous system?
Well, they're involved in a whole bunch of processes that are essential for the nervous system to develop and function. Like during embryonic development, RTKs help guide the migration of neurons, making sure they end up in the right spot.
Okay.
They're also involved in the formation of synapses, those connections between neurons.
So they're helping to build those connections in the brain.
That's a great way to think about it. And it doesn't stop there. RTKs are also involved in synaptic plasticity. That's the ability of synapses to strengthen or weaken over time.
Which is how we learn and form memories.
Exactly. One example that's been well studied is the TRKB receptor.
Okay.
TRKB is activated by BDNF, brain-derived neurotrophic factor, which is released from neurons when they're active.
So when neurons are firing together, they release BDNF, which then activates TRKB receptors on nearby neurons, strengthening the connection between them.
Precisely. That process is thought to be critical for long-term potentiation, which is how we form memories.
It's incredible that these tiny molecular events can lead to something as big as memories.
It is. And the role of RTKs in the nervous system goes even further. They're actually involved in a lot of neurological disorders, too.
Oh, really?
Yes, things like Alzheimer's and Parkinson's.
So if something goes wrong with those receptors, maybe because of mutations or environmental factors, it could contribute to those diseases.
Yeah, that's a major area of research right now. Scientists are trying to figure out exactly how changes in RTK signaling might contribute to neurons dying or not working properly.
So if we can understand how RTKs are involved in those diseases, maybe we could develop new therapies.
That's the hope. Researchers are exploring different ways to change RTK activity in the brain. Maybe by increasing signaling to keep neurons alive, or blocking signaling to prevent them from dying.
So it's like we're trying to fine-tune the signal.
Exactly. Now, there are definitely challenges in developing drugs that can target RTKs in the brain, but the potential benefits are huge.
This has been a really interesting look at RTKs in the nervous system. Before we wrap up, any key takeaways for our listeners?
Well, I think the main point is that RTKs are involved in way more than just cell growth. They play a much bigger role than we originally thought.
Right.
They're essential for our nervous systems to develop and function, and when they go wrong, they can contribute to serious diseases.
It's amazing how these receptors are involved in so many different things, from cell division to memory formation.
Absolutely. And as we learn more about RTK signaling, we're likely to find even more amazing roles that they play.
This has been a fantastic deep dive into the world of receptor tyrosine kinases. We've covered so much, from how they work to their roles in health and disease, and even the potential for new therapies.
We started by talking about how these receptors are involved in cell growth and survival. Then we looked at their roles in the nervous system and even how they can be involved in neurological disorders.
We talked about BCR-ABL inhibitors and how they've revolutionized cancer treatment, and we even looked ahead to the future of RTK-based therapies.
It's been an amazing journey through the world of cells and molecules, and it really highlights how powerful scientific research and innovation can be.
I'm feeling inspired by how complex and beautiful these processes are.
Me too. It's incredible to think about all the different signals and feedback loops that are happening constantly in our bodies to keep us alive and functioning.
I'm really excited to see what the future holds for this field.
I agree. The potential is enormous.
Well, on that note, I think it's time to wrap up our deep dive into RTKs.
Until next time, keep learning and stay curious.
To all our listeners, thanks for joining us. We hope you enjoyed this deep dive, and we look forward to exploring more fascinating topics with you in the future.